Hello everyone, my name is Walter, and I'm a PhD student at Cambridge University. I'm in the Bioelectronic Systems Technologies group headed by Professor Rasheen Owens. And today I want to talk to you about our developments of a bioelectronic platform for investigating the role of extracellular vesicles in cancer metastasis. Cancer metastasis is a process whereby primary cancer cells colonize other parts of the body and form additional tumors. The epithelial to mesenchymal transition might contribute to cancer metastasis by driving invasion. Now, epithelial cells are characterized by tight junctions, which form continuous intercellular barriers between the cells. Now, this barrier separates tissue spaces and regulates selective movements of solutes. So EMT at primary tumor sites will allow the cancer cells to separate from neighboring cancer cells and acquire a mobile and invasive phenotype, and this allows them to spread in the body. Extracellular vesicles, or EVs, are phospholipid bilayer delimited particles of varying size and composition secreted by cells. EV is a collective term that covers a range of heterogeneous subtypes of generally two subcellular origins, as depicted here. The key functional role of EVs is associated with intracellular communication, where they mediate the transfer of active biomolecules between cells, both in the vicinity of the source cell and at distant sites, to elicit a variety of phenotypic responses. The surface compositions of EVs determine their specificity and facilitate their uptake in recipient cells. Now, it has been shown that EVs from cancer cells promote an EMT-like process in pre-malignant epithelial cells. Once we have our isolated EVs, we use various methods to characterize them. This is important because we need to ensure that we indeed do have EVs, but we also need to define their size and composition. So using nanoparticle tracking analysis, we get a size distribution of our particles as well as a concentration. We also look for certain protein markers that we expect to be enriched in our EV isolate. We also look for proteins that we don't expect to be present to ensure that the sample is relatively pure. Lastly, we use transmission electron microscopy to resolve single EVs. And as can be seen here, they have a characteristic cup shape, and the lipid bilayers appear as darkened areas due to the urinal acetate staining. Now that we have a defined EV sample, we can apply that to a culture of epithelial cells. In our experiments, we use MCF10 cells because they are non-malignant breast epithelial cells, and they offer an ideal model for studying EMT. After about 3 to 5 days of incubation with the cancer-derived EVs, we observe that the cells undergo a change in phenotype. So the mesenchymal phenotype is characterized by an increased migratory capacity as well as invasiveness, and as such, these cells are now able to migrate away from the epithelial layer. Taking a look at our cells through a microscope, we can see that in our control condition on the far left, the cells maintain their polygonal shape and they remain tightly packed. As a positive control on the far right, we use transforming growth factor beta to induce EMT. And here we can see that the cells show a more fibroblast-like morphology in that they are elongated and large. So in the middle, in our experimental condition, we can see that the cells treated with cancer-derived EVs have now assumed a morphology very similar to the positive control, which indicates that the EVs are inducing a change in phenotype, potentially a mesenchymal phenotype. Now, we're actually able to electrically monitor this change in the cell barrier integrity using the organic electrochemical transistor, also called the OECT. And here we have a transistor channel made of the conducting polymer P.PSS. Now, besides being optically transparent when deposited as a film, P.PSS is ideally suited for integrating biological systems, electronic sensing elements. And this is due to its biocompatibility and its conducting properties. So in the past, OECTs have been used to monitor cell layer coverage as well as cell differentiation. And by replicating our experiment from before on the OECTs, we're able to measure the change in ionic flow as a consequence of cell barrier disruption. So on the left-hand side, we have our tightly packed epithelial cells, and they create an insulating layer on top of the transistor channel. Now, once we treat our cells with the cancer-derived EVs, they undergo EMT. And once they have assumed this more mesenchymal phenotype, these tight junctions break apart and the ionic flow through the transistor channel increases. So this gives us an electrical readout of EMT.